Cancer, the emperor of all maladies, has plagued humanity for millennia. Our understanding of this complex disease has evolved dramatically over the centuries, from ancient theories of humoral imbalances to the modern era of molecular medicine. In recent decades, a revolutionary approach to cancer treatment has emerged: monoclonal antibody therapy. Monoclonal antibodies are laboratory-produced molecules engineered to mimic the immune system's ability to target and destroy specific cells. They represent a paradigm shift in cancer treatment, offering a targeted approach that minimizes damage to healthy cells. This precision medicine approach holds immense promise for improving patient outcomes and reducing the debilitating side effects often associated with traditional cancer therapies. The journey of a monoclonal antibody from the laboratory bench to the patient's bedside is a testament to the power of scientific ingenuity and collaboration. It involves a complex and meticulously orchestrated process encompassing gene cloning, protein expression, purification, conjugation, optimization, quality control, regulatory approval, and rigorous clinical trials. Each step in this intricate chain is crucial for ensuring the safety and efficacy of these life-saving therapies. This essay delves into the fascinating world of monoclonal antibody production, exploring the scientific principles and technological advancements that underpin this groundbreaking field. From the identification of target genes to the final stages of clinical testing, we will unravel the complexities of this remarkable process and its profound implications for the future of cancer treatment. The foundation of monoclonal antibody production lies in the identification of suitable target genes. These genes encode for specific proteins, often found on the surface of cancer cells, that act as unique identifiers, distinguishing them from healthy cells. These protein markers, known as antigens, serve as the bullseye for monoclonal antibodies, guiding them with laser-like precision to eliminate cancerous cells while sparing healthy tissue. The quest to pinpoint these target genes is a meticulous process, drawing upon the collective knowledge of geneticists, oncologists, and immunologists. Scientists scour the genetic makeup of cancer cells, comparing them to their healthy counterparts, searching for subtle but significant differences. These differences, often manifested as overexpressed genes or unique mutations, provide the molecular fingerprints that differentiate cancerous cells from their normal counterparts. Once a promising target gene is identified, the next step is to isolate and amplify it. This is achieved through a process called gene cloning, where the desired gene is inserted into a carrier molecule known as a vector. This vector, often a circular piece of DNA called a plasmid, acts as a molecular courier, delivering the target gene into a host cell, typically a bacterium or a yeast cell. The host cell, now transformed with the target gene, becomes a miniature factory, churning out vast quantities of the desired protein. This process, known as protein expression, is the cornerstone of monoclonal antibody production, providing the raw material for these targeted therapies. The efficiency of protein expression is crucial, and scientists employ a variety of techniques to maximize the yield of target proteins, paving the way for the next critical stage: protein purification. With the target gene successfully cloned and inserted into a suitable host cell, the stage is set for large-scale protein production. This critical step takes place within specialized vessels known as bioreactors, which provide a controlled and optimized environment for cell growth and protein expression. Bioreactors, ranging in size from small laboratory-scale units to massive industrial fermenters, are carefully designed to mimic the physiological conditions required for optimal cell growth. Temperature, pH, oxygen levels, and nutrient supply are meticulously monitored and adjusted, ensuring that the host cells thrive and produce the desired proteins at maximum efficiency. The choice of host cell is crucial and depends on various factors, including the complexity of the target protein, the desired yield, and the intended application of the monoclonal antibody. Bacterial cells, such as Escherichia coli, are often favored for their rapid growth rates and ease of genetic manipulation. However, for more complex proteins requiring specific post-translational modifications, mammalian cells such as Chinese hamster ovary (CHO) cells may be preferred. the bioreactor becomes a bustling metropolis of microscopic factories, each cell diligently producing the target protein. This process, however, is not without its challenges. Scientists must constantly monitor cell growth and protein expression, optimizing conditions to maximize yield and ensure the quality of the final product. This delicate balancing act requires expertise in cell culture techniques, protein chemistry, and bioprocess engineering. Section 4. Refining the elixir protein purification techniques. As the bioreactor hums with activity, the next critical step is to isolate and purify the desired monoclonal antibodies from the complex mixture. This purification process requires a series of sophisticated techniques to separate the therapeutic gold from the biological chaff. The first step is harvesting the cells from the bioreactor. This involves separating the cells from the spent culture media, typically through centrifugation or filtration. Once harvested, the cells are lysed to release their intracellular contents, including the precious monoclonal antibodies. The crude cell lysate undergoes a series of purification steps to remove impurities and concentrate the desired antibodies. These steps may include precipitation, filtration, and various chromatographic techniques. One of the most powerful methods is affinity chromatography. This technique exploits the specific binding affinity of the monoclonal antibody for its target antigen. The antigen is immobilized on a solid support, creating a molecular fishing net that selectively captures the desired antibodies. Additional steps, such as ion exchange chromatography or size exclusion chromatography, may be employed to further polish the antibody preparation. The final product is then meticulously characterized and tested for its biological activity. Section 5. Precision engineering antibody conjugation for targeted therapy. In the realm of cancer treatment, precision is paramount. While monoclonal antibodies possess an inherent ability to recognize and bind to specific antigens on cancer cells, their therapeutic efficacy can be significantly enhanced by conjugating them to other molecules such as cytotoxic drugs, radioactive isotopes, or toxins. This process, known as antibody conjugation, transforms these biological missiles into guided warheads, delivering their lethal payloads directly to the heart of cancerous cells. Antibody conjugation is a delicate balancing act, requiring expertise in protein chemistry, linker technology, and pharmaceutical sciences. The choice of linker molecule is critical as it must stably attach the cytotoxic agent to the antibody while preserving the antibody's ability to bind to its target antigen. Various conjugation chemistries have been developed, each with its advantages and limitations. Some linkers are cleavable, designed to release the cytotoxic payload only upon reaching the tumor microenvironment, minimizing off-target effects. Others are designed for stable conjugation, ensuring that the cytotoxic agent remains attached to the antibody throughout its journey in the body. The conjugation process must be carefully optimized to ensure that the antibody retains its binding affinity and specificity for the target antigen. Excessive conjugation can hinder the antibody's ability to recognize and bind to its target, rendering it ineffective. Conversely, insufficient conjugation may result in suboptimal drug delivery, reducing the therapeutic efficacy. Section 6. The quest for perfection, optimizing antibody, binding affinity and specificity. The effectiveness of monoclonal antibody therapy hinges on the exquisite specificity and high binding affinity of antibodies for their target antigens. This selective targeting is what sets these therapies apart from traditional chemotherapy, minimizing damage to healthy cells and reducing debilitating side effects. However, the journey from identifying a target antigen to developing a highly effective monoclonal antibody is rarely straightforward. Natural antibodies, while possessing inherent specificity, often exhibit suboptimal binding affinities for therapeutic applications. This is where the ingenuity of antibody engineering comes into play, employing a range of techniques to enhance both the affinity and specificity of these biological missiles. One approach involves generating vast libraries of antibody variants, each with subtle differences in their antigen binding regions. These libraries, created through techniques such as phage display or yeast display, are then screened for variants exhibiting superior binding characteristics. Another strategy focuses on modifying the amino acid sequence of the antibody's antigen binding region through techniques like site-directed mutagenesis. By introducing specific mutations, scientists can fine-tune the antibody's binding affinity and specificity, enhancing its ability to discriminate between target and non-target cells. The optimization process is iterative, involving a constant cycle of engineering, expression, purification, characterization, and testing. Each iteration brings the antibody one step closer to achieving the optimal balance of high binding affinity and exquisite specificity, maximizing its therapeutic potential while minimizing off-target effects.